Hi, today we'll be talking about nutrition in plants. And for this chapter in nutrition in plants, there are a few things you need to take notice of. Okay, so today we'll be breaking down our lesson into two main parts. What exactly is photosynthesis, and basically how the structure of the leaf, the internal and external structure part of the leaf will help to carry out the function of photosynthesis. The organelle that is required for photosynthesis to occur is the chloroplast. And within the chloroplast, you actually contains the green pigment, which is chlorophyll. Okay? So these are the things that will be tested for you in exam and you are required to know. So at the end of the day, you will number one need to know what are the conditions that are required for photosynthesis. This can be found in the word and chemical equations of photosynthesis. For pure bio students, you will need to know the word and chemical equation for photosynthesis. For science bio students taking O level and N level, you will just need to know the word equation. Okay, and do take note, we usually refer sunlight as light energy. Do not write the word sunlight. It is not likely to be accepted in O level. It's better to use the actual key term light energy. Okay, so first of all, we will ask the question, where do plants actually need to eat, and where do they actually get their food supply? So generally, right, we are talking about how the plants are able to manufacture food. So this will actually come into the understanding that what are the organelles required. So like I said, plants they have this organelle called the chloroplast, and the chloroplast, like what you have studied in the chapter of cells, they actually are the ones responsible to convert light energy into organic materials for the plants to gain food. And along this chapter, you also realize what are the other items that are needed. Some of them are water and carbon dioxide gas. Okay, so these are the few main things that are required for photosynthesis. And while we look at the conditions necessary, we are also able to tell that these are the conditions, right, that would actually, once you influence the concentration, would actually affect the rate of photosynthesis. So for this particular one, we are actually looking into carbon dioxide. We are looking into chlorophyll, which is actually more of like quantifiable by the organelle chloroplast. And lastly, we will focus more on the water and not so much on the mineral salts. So first of all, while we look into these three particular conditions, right, we also ask ourselves, as a scientist, when we don't even know what are the conditions, are we able to validate, are we able to test whether these conditions are required? So there are a few experiments which you can actually take notice of, and these are examples of questions that can come out under structure or MCQ, where you are required to also understand what are the results of these experiments to decide what kind of conditions is required for photosynthesis to occur. So number one is, first of all, for any experiment, the first step is to actually destarch the plant. What is destarching the plant? Destarching the plant is basically you stopping the plant from undergoing further photosynthesis, which will lead to the accumulation or increase in the number of carbohydrates, or mainly glucose and starch in the plant. So what happens is, in order to ensure that the plant has utilized all the carbohydrates stored in itself, it is actually placed in a dark room for 2 days. Okay, and in this case, during this period of time, the plant can actually undergo respiration to utilize all those carbohydrates that are actually stored in the plants, and then utilize it for such that the concentration of starch would actually be, or carbohydrates in the plants will actually be very low. And in this case, and when you actually test it out, right, it's more or less validated to see whether the condition that you're experimenting is required for photosynthesis. Okay, so in exam, if they ever ask you Why is a potted plant destarched? It's mainly to actually to remove any form of carbohydrates that is actually manufactured before the start of the experiment due to photosynthesis, and putting it in a dark room will then allow it to actually remove all these carbohydrates via respiration. Hence you can actually then start to test the potted plant. Okay, next, a leaf is removed and test for the presence of starch. Okay, so this one is mainly to act as the control. Okay, so this one is to actually see to see whether, right, the condition that after the discharge, right, is there still starch produced. So what you should see is there shouldn't be the presence of starch because photosynthesis did not occur due to the presence of light energy. Because in a dark room there is no light energy, so photosynthesis cannot occur. As photosynthesis cannot occur, no glucose will be produced. No glucose is produced, no glucose will be converted to starch. Okay, so that's point number 2. Number 3 is using another leaf, usually sandwiched between two pieces of dark paper. And this is actually placed in strong sunlight. So we are talking about number 2 and number 3 as two separate things. Number 2 and number 3 are two separate leaves. So the other leaf, right, what will happen is it is being covered by this black paper. Why black? Because we realize that black is actually one of the better few colors that can actually prevent light energy from penetrating it, and therefore what you will expect is the areas that are covered black, right, shouldn't undergo photosynthesis. So no photosynthesis should have occurred, whereas the area that is left exposed, there is actually exposed of light energy, and hence if let's say light is responsible for photosynthesis, you should be able to test for the presence of starch. Okay, so this is one comparison that you can actually do. So the leaf is then tested for the presence of starch after a few hours, and this is the observation. What we realize is that the green area, right, that is exposed to the light energy is the one, this color is a bit confusing, but I repeat myself. So the area where there's the star shape, right, is actually green in color. Okay, and before that and after it has been tested for starch, what will happen is this area turns blue-black. Why it turns blue-black? It turns blue-black to indicate the presence of starch. Okay, and because it's actually tested and presence of starch, this will actually indicate that photosynthesis has occurred. But do take note that photosynthesis do not produce starch. Photosynthesis indirectly produces starch. Photosynthesis actually produces glucose first. So this is one main misconception a lot of students actually have. Second thing, the area that is covered black in color, right, will actually remain yellow. So this green area, right, is not exposed to light energy. So this one is light energy exposed. Okay, so since it's not exposed to light energy, this one when test with iodine, it should remain brown, or you can say remain yellow, whichever is good for you, and hence photosynthesis did not occur. Okay, so this is how you actually do the comparison, and only the parts that is exposed to the sunlight, or what we call light energy, it will be stained blue-black, as light energy is required for photosynthesis. So like what I say, try not to use the word sunlight, try to use the word light energy. It's a better way of rephrasing. Okay, so then again, the first one is to check for the condition of light energy. So second one is we are trying to see if chlorophyll is necessary for photosynthesis. So we try to narrow down chlorophyll in the form of green pigment. But do take note, chlorophyll comes in many different color. Okay, but in most of your plant is generally in green color. So what we use is actually we call variegated leaf. So what is variegated leaf? Variegated leaf, right, is basically a type of leaf that contains spots or that contains area without chlorophyll. So how is it shown? It's generally one piece of leaf where you will see many different white spots or lighter shades of spots, then the rest is actually green in color. Okay, so generally that's the one. And do take note that same step again, you destarch it. That's the first step. After that, you subject the plant to strong sunlight, because the area where the leaf is already no chlorophyll, right, is generally white in color, whereas areas that are green in color, that's the indicator where chlorophyll is actually present. So after that, you just test for starch. So what will happen is, if you can see this particular image here, right, you will see that the area that is green in color, right, contains chlorophyll, and this would actually turn blue-black. This area will turn blue-black. Why will it turn blue-black? Because starch is present. As if starch is present, photosynthesis has occurred. However, for the area which is the white in color, or let me highlight this as the yellow one, the yellow area here, these are the area, right, that does not contain chlorophyll. And if it does not contain chlorophyll, starch is absent. And how do you know starch is absent? The area will remain brown or yellow. And if the areas remain brown or yellow, this will indicate that photosynthesis did not occur. Okay, so this is generally the idea of this experiment. So next we test the third condition, is whether carbon dioxide is necessary for photosynthesis. So you can see through this experiment that there are some area where you actually use different kinds of control setup. So a control setup is basically an area where it's based on a normal environmental condition, whereas experimental setup is whereby the area, right, is maybe lack of a particular condition, or you actually adjust one of the condition to do a comparison. The reason why we want to do a control is so that we can actually validate and to compare that the condition that we have changed does really affect the results. In this case, whether carbon dioxide really do influence the rate of photosynthesis. So again, we destarch the plant. Okay, and then we use polythene bag. Now, why do we use polythene bag? Why do we cover? This one can come out as an exam question for structure. It's because inside your soil, right, it contains a lot of microorganisms. Now, what are these microorganisms? It can be bacteria and things like this, and they undergo photosynthesis. They undergo respiration. And if they undergo respiration, they would actually produce CO2. There will be an increase in the CO2 level. So this would then influence the experimental condition, because we are trying to control in one area where there is no carbon dioxide. CO2 is carbon dioxide. No CO2, and over here the control you have CO2. This is a tick. Okay, so what we can see is we cover it so that we can't count the number of microorganisms. So we just ensure that the carbon dioxide within the polythene bag will stay inside the bag and not be affected inside the bell jar. Okay. Second thing is we actually have potassium hydroxide solution and soda lime. So soda lime is there to absorb your CO2. Why? Because gas is also moving in, like oxygen gas nitrogen gas, so many gas is moving in and out. So while like photosynthesis, you always produce carbon dioxide, you always produce oxygen gas. So what will happen is oxygen gas will also be removed out of the bell jar, and if let's say you put it in a container, a fixed container, a closed one, then there will be an increase in pressure within the glass jar, and then it can also break. But of course this will not happen because the gas produced is very little or negligible. But in this case, we put the soda lime with the opening to allow air to come in and out, but only no CO2 comes in. That means it absorbs CO2, so there is no CO2 entering into the bell jar as compared to the pebble. Okay. Potassium hydroxide solution is actually used more towards to test for the presence of carbon dioxide gas. Okay, and when so generally not just to test but I'm sorry, it's not so much of testing, but actually it is to absorb your carbon dioxide gas. If let's say any actually goes in, right, carbon dioxide gas is actually being absorbed by your potassium hydroxide solution, as compared to this one whereby you use water as a form of no water doesn't actually react with it. Potassium hydroxide solution is actually a very strong base, and in chemistry you will learn that for carbon dioxide is actually an acidic oxide. So what happens is a base can react with an acid in the process of neutralization. So what will happen is in this case, right, if you are interested to know the chemical equation, right, is that potassium hydroxide when react with your carbon dioxide gas will actually produce your potassium carbonate with water. Of course, if I were to balance, it will be like this. Yeah. So this is the reaction that occurs for potassium hydroxide. So in other words, right, in this case, right, if there is no CO2 being absorbed in, right, starch shouldn't be present in the leaf when you test for it. So after a few hours, you remove the leaf from each pot and then test for starch. So if let's say there is no CO2, okay, there will be no photosynthesis occurring. And if there is no photosynthesis, starch is not produced because of the glucose is not produced, and therefore it should remain blue black. Sorry, it's not blue black, it will remain yellow. Whereas if there is in the control CO2 is present, okay, as CO2 is present, photosynthesis will occur. And when photosynthesis will occur, glucose is produced, resulting in starch is actually present. And when starch is present, it will actually turn blue-black. Okay? Yep. So pay no attention to the color of the leaf. Okay, so the iodine turns blue-black for the leaf exposed to the air with carbon dioxide gas. And in this case, the experiment do not have CO2. So generally, this one is actually wrong. Okay? It shouldn't be this color. Okay, so do take note of it. So after so many experiments, we have actually more or less checked that these are the conditions required for photosynthesis. Now, I know you all never do the experiment for suitable temperature and water. But what is these two mainly for, or what do we want for temperature? Suitable temperature because for photosynthesis, it involves enzyme. And like we studied in the previous chapter, you can go and watched my previous videos on enzymes, enzymes are actually influenced, the rate of enzymes activity is actually influenced by either temperature or pH. So in this case, it involves enzyme and therefore it is being influenced by the temperature. Having the three different stages at low temperature, at the optimum temperature, in between there is an increase of temperature, and of course temperature above the optimum temperature. So what happens is in this stage, right, the enzymes will denature, and when the enzymes denature, there is a reduction or no photosynthesis. Okay, maybe I'll put X. Yeah, no photosynthesis. Optimum temperature is whereby you have the fastest rate of photosynthesis with respect to temperature. Okay, then of course low temperature is basically very low rate of photosynthesis because of the kinetic particle theory. So what is photosynthesis? Photosynthesis is then the process in which you actually use light energy absorbed by chlorophyll, which is from the chloroplast, to convert it into chemical energy. Chemical energy is in a way your glucose. So it requires inorganic materials like your carbon dioxide, water to form glucose. Why glucose? Because glucose, right, like what we have studied so far in the previous chapter, glucose is C6H12O6. This is the chemical formula of glucose. Generally you can see it is organic in nature because it contains a lot of carbon atoms. Okay, and then the carbon comes from where? It comes from the carbon dioxide. So the carbon atom from carbon dioxide provides the carbon material for glucose. Then of course the O2 from carbon dioxide and also the H2O from water forms the other. And this conversion can only occur with the help of light energy and chlorophyll. So where does photosynthesis usually occur? Photosynthesis usually occur in the leaf of the plant. Not so much in the roots, because the roots, right, are covered in soil. And when it's covered in soil, right, the light is not able to penetrate through, the sunlight is not able to penetrate through the soil, so your roots will not be able to absorb any form of light energy. Hence, your root hair cells, they also do not contain chloroplast, so you cannot undergo photosynthesis. However, the green leaf, right, contains chloroplast, which then contains the green pigment chlorophyll, and these are the ones responsible for photosynthesis. Okay, later we will study that there's two main layers where photosynthesis can occur. Generally the palisade mesophyll cells and the spongy mesophyll cells layer. Now photosynthesis can be broken down into two main stages: your light dependent stage and your light independent stage. Light dependent means you use light, you need light. Like you are dependent on something. So generally when you are dependent on something, that means you need that thing. On the other hand, light independent means you do not need. You are not dependent on the variable, and in this case we are talking about light. So the light stage is light dependent, then of course there's a dark stage. But together is the whole thing together is photosynthesis. So in exam, if they ever ask you whether you need light energy for photosynthesis, your answer is still yes, because you will still need light energy for the light dependent stage. Okay, this came out as I think one of the essay question in a O-level question. So do take note on how to explain. Okay, so what happens in the light dependent stage? In the light dependent stage, there is the absorption of light energy by chlorophyll. That's the first part. After that, there is also the conversion of this light energy into chemical energy, and the photolysis of water. What is meant by photolysis? Photolysis means you use light. You use light to break down water. To form oxygen and hydrogen atoms. Okay? So in this case, this is already given to you at the equation. So however, in the light independent stage or in the dark stage, these hydrogen atoms will then combine together, okay, with CO2, or not so much on combine together. Hydrogen atoms, right, that are being produced from your initial stage of the photolysis of water helps to reduce CO2. So there's an oxidation step and a reduction step in chemical equation that you will learn in maybe in chemistry, but in bio you just need to take note that these hydrogen atoms are important to help to reduce carbon dioxide gas. That means carbon dioxide is being reduced by hydrogen atoms to form your glucose and water. So this whole thing is controlled by a series of enzyme reaction. The whole steps of the series of enzyme reaction is not going to be tested for your O level. You just need to know that it is actually to reach carbon dioxide gas to become glucose and water, it requires the use of the hydrogen and enzymes. That's all. Okay, and this is the dark stage or the light independent stage. So overall, please go and memorize this. This one is very important. You will need to know the word and chemical equation. The one at the top is actually your chemical equation. And then the one at the bottom is actually known as your word equation. In exam, do take note that which are they want. Do they want the chemical or do they want the word? As I say, only pure bio students taking the 6093 syllabus, you are required to know both, in which either one will come out. Most probably is the chemical one. Whereas for students taking science bio, O level and N level, you will just need to know the word equation. Okay, and do remember to also write in between the arrow the condition, which is light energy and chlorophyll. Do not go and write things like carbon dioxide plus water plus light energy plus chlorophyll to give you glucose. Okay, this method of writing is wrong. It should be written at the top of the arrow and below. Second thing is, do take note that it is actually glucose that is being produced and not starch. So what you are testing, right, is not the glucose, you are testing the starch. So what happens is there will be one more step where the glucose is converted to starch. But how the glucose convert to starch, no need to know. You just need to know that for photosynthesis, you produce glucose. One common mistake is a lot of students tend to replace glucose with starch. Okay. There are some factors that will actually affect the rate of photosynthesis. I would urge you to remember the shape of the graph. And this usually comes out in MCQ. So when we look at the shape of the graph, like for example, light intensity, what happens is when light increase, when the light intensity increase here, okay, as you can see, as light increase, the intensity, rate of photosynthesis will also increase. But then it will only increase to a certain point. Okay. Now, this is what we call the limiting point. Or why do we say that? Is because at this particular point, any increase in light intensity do not influence the rate of photosynthesis. So there must be other factors that actually affect photosynthesis. So we can say that in this region here, light is actually the limiting factor. What is meant by limiting factor? Limiting factor is basically a condition that would actually influence the rate of photosynthesis. It will limit the rate of photosynthesis. It will control the rate of photosynthesis. But as you can see, any point after this area here, the mark X, light is no longer a limiting factor. And when light is no longer a limiting factor, but you can see that light photosynthesis is still occurring, that means if any other further increase, it is due to other factors influencing rate of photosynthesis. Now, how is this so? What are the other factors? You have carbon dioxide concentration, you have temperature. So these are the ones. So same thing for carbon dioxide gas. Until this point here where it starts to plateau, CO2 is the limiting factor. Whereas anything above here, after this point, CO2 is no longer the limiting factor. Other factors are the limiting factor. Okay, it can be light intensity, it can be temperature. Now this shape follows the enzyme curve. But even this shape is also slightly wrong. How should the shape of the enzyme curve look like? How should the rate should look like? It wouldn't be a very nice bell curve. Instead, okay, the rate, how it will look like, right, is like I say, a very gradual increase, then a sharp decrease. Okay, there should be a sharp decrease where in the top is actually the optimum rate, and if you draw a line down, it is where the optimum temperature is at. So do take note of that. Okay, so definition of limiting factor, you also need to know. A limiting factor is a factor that directly affect or limits a process if it is a quantity, if its quantity or concentration is altered. Okay, please do not use the word amount. Please use words like concentration, mass, volume to quantify things in exam. Okay, now some of you may ask, so how can then why is then the any increase in light intensity don't affect the rate of photosynthesis? Reason being is because in a leaf there is a X number of chlorophyll. You won't see suddenly as light increase there will be more chlorophyll, you see. So what is then hindering the light intensity is actually the number of chloroplasts or chlorophyll in the plant. Of course, having more chloroplasts results in more light intensity being able to be absorbed, and hence rate of photosynthesis can occur. For carbon dioxide, concentration is then also limited by the number of stomata pores for gas to come in and come out of the plant. So these are the things that actually, if let's say they ask you a further question on what affects the light intensity as a limiting factor, for light intensity it will be the number of chloroplasts. For carbon dioxide it will be the number of stomata pores. Okay, so what is the effect of light intensity on the rate of photosynthesis? This is one apparatus which we actually use. This has came out in science biology practical examinations for O-level a few times already, where you have to do the setup. Otherwise, I've seen it more or less coming out as a structured question in schools exam papers during prelim, mid-year exams, or end of year exams. Okay, otherwise this will also can become a question where you can actually plot a graph. So this is a very nice questionnaire. So then how do you then understand the effects of light intensity on the rate of photosynthesis? Number one, let's understand the different apparatus. Having a water bath to keep temperature constant is to actually make sure that it is always at an optimum temperature for your enzymes in photosynthesis. That means you will always have the optimum rate, correct? Second thing is having the water plant acts to produce you need a plant, right, to undergo photosynthesis, and this lamp Later you'll realize by adjusting the distance, you are controlling the light intensity. How are you controlling the light intensity? Number one is closer means higher light, or closer to plant means higher light intensity. Whereas further away from plant means lower light intensity. Okay? So these air bubbles are actually given off at the end of the reaction, and sometimes allow for the rate of bubbling to stabilize. So what you can do is you usually count the number of bubbles over a period of time, and then you count the rate. Okay, we are not just counting the numbers, we are counting the number of bubbles over a period of time. Okay, and this will allow rate. And of course you repeat the experiment by moving the light source closer to the plant, and hence this would actually indirectly increase the light intensity. Then you repeat the reading again. And it is then observed that the rate of bubbling will increase as the decreasing the distance of the lamp from the plant decrease. Now what does this mean? If you want to apply your concept, is that you know that rate of bubbling increase. This could infer that rate of photosynthesis increase. Okay, and it's increased due to the increase in distance of the lamp to the plant, correct? It's a decrease. And this means that the light intensity is higher. So what this means is that higher light intensity results in higher rate of photosynthesis. Okay, why? Because the bubbling, if you want to guess what is the gas that is given out for the air bubbles in photosynthesis, it's actually oxygen gas. Oxygen gas is being produced. Now there is one experimental error for this. What is this experimental error? Is that you cannot. Okay, what is experimental error? Experimental error is an error that is due to the experiment itself and not because of your human error. Okay, so an experimental error is because that you cannot control the volume of gas produced in each bubble. So what this means is that the air bubble that is produced, right, some of it is of different sizes, and of different sizes will have different volumes of carbon dioxide gas. So sometimes if you have very small bubbles but you have a lot of number of bubbles, this doesn't mean that it has a higher rate of photosynthesis as compared to one experiment where you've given out bigger bubbles but lesser number of bubbles. Okay, so how I would then try to correct this experimental error, right, is a better way is to measure the volume of oxygen gas being produced. And then I measure the rate. Then you measure the rate. And how you do it is basically you put maybe a gas syringe over here. So you put one gas syringe over at this area here. So that what will happen is the gas will actually increase, will push the plunger up, and therefore you will be able to measure the volume of oxygen being produced. But of course, do take note that you also need to ensure that there always will be a continuous supply of oxygen for the plant. Continuous supply of carbon dioxide for the plant to ensure that photosynthesis will always occur. Okay, so in this case, if we look back into the graph, you'll see that beyond point X, right, the rate of photosynthesis will remain the same even though the light intensity occur. in the experiment you wouldn't see rate of photosynthesis. Instead, what you will see is the rate of bubble production. So this one is an example of how you can go about drawing the graph. So your graph should look something like this, like it shouldn't go downwards. And and therefore we can say that light intensity is the limiting factor for the first part of the graph, and beyond x, light is no longer the limiting factor, it's some other factor. Okay. So now we look at temperature. So rather than control, so just now we were talking about light, right? So now if we look at temperature, right, the variable that we need to keep constant, right, is the distance. So that we control the light intensity. So this light intensity is actually fixed. What you vary the temperature of the water bath. Okay, so in this case, you try maybe low water, using ice cold water, and then later you just repeat the experiment. You count the number of bubbles, divide by over a period of time to get the rate. You vary in different temperature. Various temperature, it can be maybe every 10 degrees Celsius. Actually, it would be better to do in periods of 5 degrees Celsius, so on and so forth. So it can be how can this temperature be adjusted then? You either use a Bunsen burner, you use normal tap water or distilled water, then you adjust hot water to increase temperature, or you use distilled water to reduce the temperature. Okay, you can even use ice cube to reduce the temperature. So what you can see is, as temperature increase, okay, the enzymes will actually react again at an optimum area, and therefore rate of bubbling increase. And as the rate of bubbling increase, your rate of photosynthesis also increase. But there is a danger to this. Any temperature that is too high will lead to a decrease in the rate of photosynthesis, or a decrease in the number of bubbles formed over a period of time, because the enzymes has denatured. Okay, so do take note of that one. So in this case, if we look at it over here, temperature as a limiting factor. Okay, over here we can actually compare different kinds of environment. So first thing is we want to see whether temperature can influence. So we will compare A and B. Why? Because concentration of the CO2 is actually the same. Then of course, if you want to compare in terms of temperature, you can also use graph C and D, but only between C and D, because the temperature is also the carbon dioxide is also the same. So generally you will see that the graph, right, is actually higher at higher temperature. This means that the rate is higher at higher temperature. Now, what happens if then you want to compare otherwise? So if let's say you want to compare, so I write here, compare AB or CD, you could say that increase temperature, increase rate of photosynthesis. However, if you look at, so this one is, CO2 is the same. So now if you want to compare CO2, right, there's also light here. So in this case, we will assume that the light is consistent through every single period. Actually it's consistent throughout every single period. So what will happen is you can compare A with C or B with D for temperature being constant. With temperature being constant, you can compare whether the concentration of CO2 affect. So the higher concentration of CO2, the higher the rate of photosynthesis. Now what we can see over here, right, the area where I mark the X, right, these are areas where actually light stops becoming the limiting factor, because you can see the graph starts to plateau. So that's how you also tell. Okay. So the effects of increasing temperature on the enzymes. So what will happen is, because in the light independent stage, enzymes are required to contribute to photosynthesis. So the reactions in photosynthesis are enzyme dependent, and at low temperature, enzymes are inactive. At extreme temperature, more than or higher than the optimum temperature, the enzymes will denature. And when enzymes denature, you will not form the enzyme substrate complex. Okay, no enzyme substrate complex form, and if there is no enzyme substrate complex form, there is no rate of enzyme reaction, therefore no photosynthesis occurring. Okay. So this one is basically, again, very straightforwardly explained. So you can say it's at low temperature, okay, low kinetic energy. Having low kinetic energy, you have lesser frequency of effective collision between the enzyme and the reactants for photosynthesis. Therefore, lower enzyme substrate complex form, then hence lower rate of photosynthesis. Okay, I'm talking about rate here. Whereas as temperature increase, you gain in kinetic energy. This increase the frequency of effective collision, and therefore it increases the rate of photosynthesis. At the top, you will see is at optimum temperature. So this is the maximum number of enzyme substrate complex form. Therefore, optimum, or you can say maximum rate of photosynthesis. As temperature increase beyond the optimum temperature, okay, the enzymes denature. So in other words, the enzyme decrease. This will mean that you have lesser enzyme substrate complex, and therefore decrease the rate of photosynthesis. Okay, so this is what happens for these 4 different conditions. Then next is, what is the effect of carbon dioxide then? So in order to adjust the carbon dioxide concentration of photosynthesis, okay, so this one is mainly for temperature, your water bath. Distance of lamp is mainly for light intensity. So it's the concentration of sodium carbonate, hydrogen carbonate, that is for CO2 concentration. Okay, so everything you need to keep the same. So say I would say constant because for this one you are talking about carbon dioxide gas, so must be constant. This one also must be constant, and this one is the one that you vary. Okay. So same thing again, the experiment is carried at room temperature. Room temperature is considered as the optimum temperature. Okay, and of course different concentrations of sodium hydrogen carbonate solutions. This means that you see the CO2 is varied, and therefore you repeat the same thing. You measure the number of bubbles over a period of time, increase concentration of sodium hydrogen carbonate solution would actually increase the CO2 concentration, hence increase bubbling rate, and therefore increase rate of photosynthesis. Okay, so generally this is how it occurs, and beyond this point, CO2 is no longer the limiting factor. CO2 is the limiting factor here. Okay, beyond here is no longer, beyond Y is no longer. And of course the rate of photosynthesis will increase when CO2 is increased. So if you are ever asked to sketch, right, do take note that if let's say the CO2 concentration increased by double, right, your height must increase by double. Double the height from here to here. Okay, so if it's double, okay, somewhere here. Sorry, I think this one is not drawn to scale. Yeah, but in other words, what the gist is, you need to know is the graph, right, must always be higher. So in exams, right, do consider, so this is like a application question where they ask you to sketch, given one as the example. Okay. So now we come to the fate of the glucose in leaves. So what will happen? So what will happen for rate of photosynthesis? In this case, right, is basically the fate of glucose in leaves is that the fate of glucose in leaves, right, they will actually be used immediately by plant cells for cellular respirations to form cellulose cell wall. So this is the one factor. The other one is this excess glucose is temporarily stored as starch. So this is the one that you test for. This is the one that you test for photosynthesis using your iodine, and of course again it's stored in sucrose to be transported in the storage organ. This one you just have to remember, it's a brain exercise kind of thing, and it usually comes out in the assay component. Okay, the fourth reason is that it can actually react with nitrates and mineral salts to form amino acids, which are then used for protein synthesis to form your new protoplasms. And of course any excess is usually carried to other areas to be storage, and of course you have the formation of fats for storage and cellular respirations to synthesize new protoplasm. Okay, so this is the first part of assay. That one usually is about 5 marks. This comes the second part. It's also another assay component in terms of the importance. So how can you actually, or why is photosynthesis important? Because plants are the only producers in the food chain, and they are the only one that is able to actually convert your chemical energy through light and convert it into chemical energy that can be stored in plants such that we can actually eat. and uh, and from there we will be able to take note. The second thing is it helps to purify air by removing carbon dioxide at the atmosphere. Basically, it helps to purify air by removing carbon dioxide from the atmosphere and then releasing it back to oxygen. So only the plants are able to do it. Then last but not least is coal are also able to be formed from trees as a form of fuel. But then this is not one of the main important reasons. It can be the main two reasons are the ones at the top. These two. Okay. And that's where we have covered for photosynthesis. Okay, in the next video I'll be covering the leaf, the internal structure of the leaf and the external structure of the leaf along with its function. So before you go, please remember to like my video, subscribe to my channel, and click the notification button so that you'll be informed of future videos that will be made available to you. If not, that is all.